So now we're going to look at these topics called synapse. So we do know that if you look at the synapse, let's say for example, okay, we have the sensory neuron. It's our body. Let's say this is the terminal of the sensory neurons. Okay, terminal of sensory neurons. Then we do have the motor neurons. So this region, do I highlight? So it's not a connected one. It's not a connected one. So means what? Means that the actual potential have to cross this gap. Right? Actual potential have to cross this gap. So this gap is known, right? Is known as synaptic cleft. Okay. So the entire structure that I highlight here is known as synapse. Okay. So an actual potential pass down an axon, eventually reach the end or the terminal here, and to all its branches. So this branch may form a junction with dendrites of the other neurons. For example, can you see that dendrite? It may form the junctions with the muscle cells or even the gland cells. So such intercellular junctions are known as synapse. Okay. So the neurons whose axon transmit actual potential to the synapse is the pre-synaptic neuron. So the first one, okay, you look at this structure. So this is the directions, right? This is the direction. So the one they're going to transmit or send over the actual potential is known as a pre-synaptic neuron before the synapse. Okay? Pre-synaptic neurons. The one that receive will be called, okay, post-synaptic neuron. Okay, pre-synaptic, post-synaptic. So later we look at this, we have the word called pre-synaptic membrane and post-synaptic membrane. So it means that's the membranes of the neurons. Okay, and the gap in between the pre and prosynaptic neurons, okay, pre and post-synaptic neuron is called synaptic cleft, right? Synaptic cleft. So it's a narrow space that separates the pre and post-synaptic neurons. So the end of the pre-synaptic axon is often, you can see that it's swollen in terms of the structure and contain many synaptic vesicles. Okay, which are each packed with a chemical known as the neurotransmitter. And because in our topic is the cholinergic synapse, so we need to know one of these neurotransmitter known as acetylcholines. Okay, acetylcholines. So this neurotransmitter is very important, even though they are very small, but it can help to transmit action potential across the synapse or even these neuromuscular junctions. Okay, so if you look at the structure and enlarge it now here, this is a pre-synaptic neuron. This is a post-synaptic neurons. In between, we have the synaptic cleft. So the entire structure is known as synapse. Okay, so the dendron, you can see that dendron, it will branch. Okay, at the terminal of the branches, you can see that they have this something like a bulb called synaptic bulb. Sometimes we call it, or synaptic knot. So this synaptic knot, if you zoom it in, you can see I've got a lot of mitochondria as well as the vesicle. So this vesicle contain the neurotransmitter. Can you see that? One example neurotransmitter that we know is the acetylcholines. Okay, acetylcholine. And then this is a pre. So the membranes at the pre-synaptic neuron is known as a pre-synaptic membrane. And we do have the post-synaptic membrane as well. But at the post-synaptic membrane, you can see that we have the receptor that on this, we call the ligand-gated sodium ion channel. Okay, so let me show you guys the whole structure. Okay, and also the process. So if you look at the structure, you can see that actually, okay, at the terminal of the axon, it forms a bulb-like structure known as synaptic knot. So this synaptic knot consists of very important three things that we need to know. First, they have a lot of mitochondria. They have a lot of this vesicle known as synaptic vesicle. Okay, and they do have a special channel protein, something like voltage gated, okay, voltage gated sodium and potassium ion channel, but now they have this voltage gated, calcium ion channel. Okay, so this one is the voltage gated, but now it's a calcium ion channel. Here, so in this vesicle, they contain a lot of these neurotransmitter. One example neurotransmitter is the acetylcholine. ACH, okay, acetylcholine in short forms, okay. Then they have a lots of these mitochondria. So the membranes, okay, the membrane is known as the pre-synaptic membrane. Okay, so this is axon, okay, the terminal of the axon terminal. Then it's synapse with the dendrite, for example, dendrite of the motor neuron. So this is the dendrite of the motor neurons. Okay, so the membrane, this is a post-synaptic neurons. So it's known as, the membrane here is known as the, sorry, guys, my mistake. So this membrane is known as the post-synaptic membrane. So on this post-synaptic membrane, very, very important, they do have the sodium ion channel. But this sodium ion channel is special. Okay, this sodium ion channel, they have the receptor. This sodium ion channel, they have the receptor. Okay, let me show you guys what is the function of this. Okay, so initially they are closed. So this structure is the ligand gated, okay, sodium ion channel. Now, what's this mean? Means that later they need to have a chemical to stimulate it to open. Ligands here means the binding or the chemical. So it means that this ligand gated sodium ion channel, so it means that we need to have a neurotransmitter that bind to this receptor, only can cause the sodium ion channel here to open at the gate. So entire story actually is pretty simple. When the actual potential arrives, because we do know that any actual potential that arrives can change the voltage. So when it changes the voltage, okay, or membrane potential at the pre-synaptic membrane here, or pre-synaptic neurons here, it can cause the voltage gated calcium ion channel to open. Okay, so first step. Okay, so first step. So arrival of actual potential. So arrival of actual potential can cause the voltage gated calcium ion channel to open. So when calcium channel open, outside actually have higher concentration of calcium ion compared to inside. So therefore, calcium ion now enter by facilitated diffusion. Are you clear? So calcium ion enter. So the influx, okay, or rapid influx of calcium ions will cause a series of reactions to take place until the exocytosis take place. Okay, so exocytosis is a what? So now, because the entry of the calcium ion channel, it will cause this vesicle that contain the acetylcholine now to move towards the cell membrane, fuse with the cell membrane, okay, and release the neurotransmitter by exocytosis. Can you see that? So when the calcium ion enter, it will cause this vesicle, right, to move towards the pre-synaptic membrane, and then the membrane of vesicle fused with the membranes of the pre-synaptic membrane, release the neurotransmitter by exocytosis. So once exocytosis take place already, then because of high concentration of neurotransmitter near to the pre-synaptic neurons and low concentration near to the post-synaptic neurons, diffusions now take place. Diffusions of the acetylcholine take place. Okay, after exocytosis, then we can see that diffusion take place. So it diffuse across the synaptic cleft, right? Diffuse across the synaptic cleft, and finally they attach to the receptor. So this receptor actually on ligand-gated sodium ion channel. Are you clear? So it means that eventually they will diffuse and then they attach. Once they attach, it will cause the sodium ion channel to open. So the openings of the sodium ion channel here, okay, due to the binding of the neurotransmitter acetylcholines, then allowed the sodium ions now to enter. So the entry of the sodium ion now depolarize, okay? So you can see that the depolarization, okay, of post-synaptic membrane. Therefore, action potential will be generated. Can you see that, guys? Understand the whole concept? Okay, so again, so first part here, calcium ion. Okay, why calcium ion enter? Because of the arrival of the action potential. So when action potential arrive at the terminal here, it will cause the voltage-gated calcium ion channel to open, allow the calcium ions to enter into the knot there, okay, or these are called the pre-synaptic knots here. And then because entry of the calcium ion, it causes the exocytosis of acetylcholine, and acetylcholine diffuse across the cleft and then bind to the receptor on this ligand-gated sodium ion channel. And because of the binding and the entering of the sodium ions, now sodium ions depolarize the post-synaptic membrane, then action potential is generated. So when action potential is generated here already, then you can see that it continues to transmit all over until they reach along the neurons until they reach the target cells. For example, the membrane, the glands. Okay, so this is how the action potential cross the synapse. Are you clear? Okay, but we need to know that the neurotransmitter cannot remain inside the synaptic cleft for a long period of time, because if I have one action potential, I want only one action potential to reach my brain. If these sodium ions continue to detach and then attach back, detach and attach back, what happens? You're going to, for one action potential, you're going to create multiple. You're going to create multiple stimulus, okay, or multiple action potential in the brain. So therefore, in order not to have this, multiple images or multiple action potential to be stimulated in our brain or arrive at our brain, we need an enzyme called acetylcholine esterase. So acetylcholine esterase is the enzyme. So we do have the enzyme known as acetylcholine, okay, esterase. So acetylcholine esterase is an enzyme to degrade or hydrolyze acetylcholine. Are you clear? So means that we want to break down the acetylcholine so that to stop the transmission. If no, you try to imagine I slap you one time, then your brain will feel like multiple times you cannot slap. Are you clear? Because why? Because without the acetylcholine esterase. Okay, so once acetylcholine esterase release or detach, when once acetylcholine detach from the receptor, then acetylcholine esterase will degrade this acetylcholine. Therefore, no more action potential will be generated until the next action potential arrive at the synaptic knot there. Okay, So understand the whole concept now, okay or not? So let us look at the diagrams here. So I repeat the process. So first, the arrival of the actual potential caused the calcium ions to enter because of voltage-gated calcium ion channel open. So the entry of the calcium ions caused the exocytosis. Okay, so the vesicle first have to move towards the cell surface membrane here, known as a pre-synaptic membrane. So fuse release the neurotransmitter by exocytosis, and then this neurotransmitter now, okay, can diffuse across the synaptic cleft and bind to the receptor. So when a neurotransmitter bind to the receptor, guys, I want you guys to cancel this potassium ion. Okay, don't talk about potassium ions. In our syllabus, we only focus at the sodium ion. So this is a ligand-gated sodium ion channel. So the binding of the neurotransmitter open the ligand-gated ion channel, sodium ion channel, allow the sodium ion to enter. So the entry of the sodium ions will cause the depolarizations of the post-synaptic neurons. Okay, so the detach, can you see that? Once the neurotransmitter detach from the receptor, then you can see that it close back. So therefore, no more depolarizations. Okay. So you can try it, the question here. So this is the answer. So you need to know acetylcholines, as I said that in our syllabus, because the synapse that we're going to use here is the cholinergic synapse. Okay, cholinergic synapse. Okay. And the enzymes, okay, they break down the acetylcholine. You need to know it's called acetylcholine esterase. Okay, hydrolysis of acetylcholine molecule. Therefore, you can actually recycle back. Okay. So what are the roles of the synapse? So very important, guys, highlight the first one. The first one is the most important one. Synapse ensure one-way transmission. Okay, so how, question always asks, how synapse ensure one-way transmission? So highlight statement B here. Because you only get neurotransmitter at the pre-synaptic neurons. You won't have the neurotransmitter at the post-synaptic membrane there. No, you don't have. Okay, so you look at the diagram again. Only the pre-synaptic neuron have the axon there, have the neurotransmitter. The dendrite, you won't see any neurotransmitter. So it means that the release of neurotransmitter always in the directions of from the pre-synaptic neuron to post-synaptic neuron. So this is the first adaptation. Second adaptation, okay, second adaptation, you can see that the ligand-gated sodium ion channel only available at the post-synaptic membrane. So therefore, neurotransmitter have to cross the synaptic cleft and bind to the receptor. So therefore, the depolarization will take place at the post-synaptic membrane. No depolarization take place. Can you see that? Because why? There is no receptor, okay, on the pre-synaptic membrane. So these are the two adaptation, okay, two adaptation. Why the synapse ensure one-way transmission. So no way that transmission of neuron can occur in the opposite direction, because you want to go through the opposite directions, cannot. Why? No neurotransmitter. I want to go here? No, because we don't have the receptor for the neurotransmitter. Okay, and also no ligand-gated sodium ion channel. Okay, now synapse allows the interconnection of nerve pathway, give us the wider range of the behavior and the responses. Now, what does this mean? If let's say without synapse, what will happen? Without a synapse, only called as a one-way route. Okay, so let's say for example, this is a sensory neuron. So sensory neuron all the way connected to our brain. So means that you can see that we only have one. For any one stimulus, you only have one response. Correct or not? Because everything goes to the brain. Do you get the idea? See? Then if no synapse, what will happen here is you touch on the object, it's hot, then you know that either it's hot, you won't remove your hand. So But it's not true, because when you touch on a hot object, few responses actually take place. First, you immediately remove, withdraw your hand. When immediately you withdraw your hand, you know that it's hot. And sometimes you see that, ouch, or you may swear. Okay, in this case, can you see that? Okay, so you touch it and then you immediately remove your hand and then you say, oh, it's hot. Oh yeah, you know it's hot. Can you see it? So multiple response. Are you clear? So for example, when you view something, okay, you look at the crisis. For example, someone take the knife, okay, or the gun want to shoot you. So if only one response, what's it mean? You see and you want to run. But you want to run involves so many muscle cells. If no synapse, how we can control so many muscle cells here? So therefore, we need to have the synapse. Okay, so synapse actually allows the interconnections of the nerve pathway, allow us to have wider range. So individual sensory and relay neurons have axons that branch to form synapse with many different neurons. Therefore, we can actually control many effectors. For example, crisis. Okay, we want to activate our adrenal gland as well to release the adrenaline. Correct or not? Can you see that? And then activate our muscle, cardiac muscle. Correct or not? We want the heart pump faster adrenaline. Can you see that? So if we don't have many synapses, then we won't be able to have many responses. Okay. Third, this is also very important. Synapse also allow integrations of the action potential. So if the depolarization of postsynaptic membrane of an intermediate neuron does not reach a threshold, therefore no action potential is sent in the neurons. Okay. Now sometimes some neurons, they are very sensitive. So when they reach, it means that we do generate the action potential all the way until the spinal cord, not the brain. Okay, let me draw it again. So all the way to the spinal cord. So at the spinal cords, we do have this intermediate neuron. So intermediate neurons here, because of the synapse, if our neuron is very sensitive, okay, then you go and pass all the image, you go and generate action potential all the way until here. Can you see that? And then you're going to cross, action potential going to cross. But the thing here is our neuron, intermediate neurons here, because of synapse, they can actually judge whether this information need to be sent to the brain or not. One of these mechanisms that we do know that, for example, okay, this is something that's always happen. So we call it sensitization. So it means that you already become immune. For example, when you work in the factory, okay, the chemical that you keep on inhale, chemical. So sometimes we go to, if you go out from the room and then enter back the room, how come I stay there 2 hours, I didn't smell anything? Are you clear? Hey, how come? It's not that you didn't smell anything. Action potential still generated, sent to the intermediate neuron, but intermediate neuron decided, oh, don't want to send to the brain because nothing, not dangerous at all. Can you see that? Are you clear? But when you move out, because you're already immune, something like immune already. So when you move out from that particular room, and then the new taste, the new smell, right? The new smell coming in. So now, oh, the new one sent to the brain to interpret. So it's something like a refresh. But when you go back to that particular room, the chemical activate again, right? Stimulate. So therefore, our synapse allow the integrations of the action potential so that your brain won't be overloaded with the sensory information. Are you clear? So some low frequency do not travel, okay, from a sensory neuron to reach the brain, so you won't overwhelm your brain, okay? And synapse also involved in the memory and the learning. Okay, this is particularly true, okay? For example, okay, if you look at the old kinds of the handphone, and not handphone, but house phone, you cannot register, okay, the phone number. You cannot register your friend's names, okay, right? So you remember the number, then you dial the number, okay? Then your phone rings, okay, your house phone. So immediately you take out the phone, by listening to the voice of your friends, immediately your brain actually picture out the face of your friends. So it means that now you can see that one is your memory through your eyesight, right? The view, register your friend's face. And then when your friends talk to you, you register the voice. But how we register? Because you see your friends and you listen to the voice at the same time. So both memory will be registered at our brain. So whenever your phone ring, even though you don't look at the context, you listen to the voice, you automatically will pick up who is that person. Why? Because the memory capture put those information all together at a particular region of your brain, then your brain actually register both information as the same information. So synapse can help us. If without a synapse, then we won't be able to control or to search the information. So that's why it's very, very important. Means we do have the synapse, because synapse are involved in the memory and learning. Okay, so we don't know how this can be formed, because brain is very, very complicated. How will our brain involved in the memory? But there is many evidence show that the memory and the learning involve the synapse. Okay, for example, your brain frequently receives information about two things at the same time. For example, the sound of a particular voice and the particular face, then it will thought to be a new synapse form to link both together. And when we think about your friend, you can recall the voice, and we listen to the voice, you can recall your friend's face. This is how, the synapse actually functions. Yeah. Normally, past year paper, you look at past year paper, question always asks for two. Okay, two rows of the synapse. The first one, you must put it in, must be this. Synapse ensure one-way transmissions, and then out of these three, you can choose either one. So normally I would say that's the second one. The more important, allow the interconnections of the nerve pathway. Are you clear? Okay. Now, how about chemical that affect? Okay, what the effect of the chemical at the synapse? Okay, so we have some chemical actually can affect our synapse, particularly nicotine. Okay, because it's very common. If you look at the Botox and also the nerve gas, they are not common, right? But nicotine is the most common one. Okay, why? Because it's inside the cigarettes. Okay, now you can always see that people say that nicotine actually increase memory. Are you clear? If you look, if you ignore the side effects on the nicotine, yes, nicotine actually improve the memory and give you much more energy. Okay, you feel like you are very energetic after you smoke. The reason here is because nicotine, the shape is quite similar to the acetylcholine, but acetylcholine will be broken down by the acetylcholine esterase, but nicotine not. Okay, nicotine is not rapidly broken down by the enzyme, therefore it remains the receptor for longer times than acetylcholine. So therefore, this nicotine will give us the effect, okay, something like, if I want to do something, so this signal sent to my muscle cell is not one, it's continuously. So you think that you are very, very energetic, but it's not true, because nicotine actually fool our muscle. Are you clear? Your brain may say that, okay, contract. Okay, contract a while, but because of nicotine, you contract more. So it means that you think that, oh yeah, my muscle can do more work. Okay, so that's not true, because nicotine cannot, or it's not rapidly broken down by the enzyme acetylcholine esterase. Are you clear? So this is the called nicotine. Okay, so but the thing is, large dose may fatal, because I will try to imagine acetylcholine, something like acetylcholine continuously remain inside, this nicotine remain inside your synaptic cleft. What will happen here is you're overfiring. So muscle contraction, we may have muscle cramp later. Okay. So next, Botox. Botox is the one that acts on the pre-synaptic membrane and prevent the release of the acetylcholine. So Botox actually is used as a medical, okay, for the muscle spasm. Sometimes because our muscle do have the problem, okay, so it continue to contract. It don't want to relax. Muscle don't want to relax. If the muscle don't want to relax, what will happen? It will use up the ATP and continue to cramps. Okay, so in this case, it's very good. Okay, we have to stop the release of the acetylcholine so that you give time for the acetylcholine esterase to remove the acetylcholine. So this is Botox. Botox is the one that you use it also as a cosmetic on the face, right? You inject. So therefore, you can see that, okay, Botox at the face there. So why? Those people with the Botox, the face is look like it's very, very firm, and because it's reduced the muscle contraction. Can I say Botox actually stops the release of the acetylcholine? Therefore, the face, it seems like no contraction. So when they smile, the smile also very fake. Why? Because they cannot control, they cannot contract their muscle. So that's why the muscle, the face muscle, look very firm and very nice shape, okay, because of the Botox. They stop or prevents the release of acetylcholine, so the face muscle cannot contract. Okay, so reduce the wrinkles. Okay. The last one is the one is quite important, okay, because we're going to learn about this nerve gas called sarin. It's used as by the terrorists as the weapon. What will happen here is the sarin gas actually act on the acetylcholine esterase. So what will happen here is it will block all the acetylcholine esterase, so the acetylcholine esterase won't be able to degrade or hydrolyze the acetylcholine. So means the acetylcholine perceive, okay, keep on, remain in the synaptic cleft and continuously produce the action potential in the post-synaptic membranes and reach the brain. So the brain will be overwhelming with many information, okay? So as if like if I slap you one time, you'll feel pain, okay? So if I touch you, you'll feel the pain like, okay, a multiple pain in your body. And then your muscle spasm because the action potential keep on firing us, okay, your body to contract. So it's a whole body to contract. And then you will be suffocated. You can't breathe because of the smooth muscle keep on contract. So this one is very severe nerve gas. Actually, a minute amount of nerve gas can kill a person already. And one of the nerve gas, sarin, have been used in the terrorist attack in few countries. Syria is one of them. Another one is in 1988 in Tokyo, where terrorists use it in the subway station. And another example is in KLIA 2, where this VX nerve gas is used to assassinate Kim Jong Nam, okay, the brother of the Kim Jong Un, okay, the North Koreans president, not mistaken. Okay, so this nerve gas actually acts very fast. Okay, they use it as a biochemical weapon here. Again, nerve gas have been used as a biochemical weapon. It's banned, totally banned already, but some countries still use it as a biochemical weapon. What they do with this nerve gas actually block the acetylcholine esterase. So it means that it will affect a lot of your synapse and also neuromuscular junction. Your muscle keep on contract, convulsion, and then because try to imagine your smooth muscle contract, so you can't breathe in the air because your cardiac muscle, your smooth muscle keep on contract. It won't relax because you breathe in and breathe out. You have intercostal muscle, right? Can you see that? So those muscle contract, so you cannot breathe, okay? Or you cannot, breathe out the air. So therefore, you can see that you're going to die painfully, okay, by using this nerve gas, okay? So any more questions from you guys? If not, then we have done for the synapse.